Thank you, Erika, for this nice introduction and for inviting me for this talk. I will tell you about our activity about food and plant imaging in the TeraHertz range at the University of Pisa. So first, let me introduce this TeraHertz region, which is quite unique because it's sandwiched between the photonics and electronics ranges. So if you work in electronics, you probably know that if you move from ultraviolet to visible, infrared, near infrared, mid infrared, and far infrared, the detection of photons become increasingly difficult and starts to be very, very difficult when you approach the 10 terahertz limit. On the other hand, if you work in electronics and you try to increase the frequency from radio waves to microwaves, and when you reach about the limit of 0.1 terahertz, electronic techniques do not work anymore. So for many years, this has been called the terahertz gap because there were no sources and detectors available. And they have been mainly developed in the new millennium. The wavelength in this region goes from 3 millimeters to 30 microns, and this gives this radiation unique properties. First of all, the energy of the photons is very, very low, lower than in the rest of the photonic range. So the operation in this region is completely safe, both for the operator and also for the samples. For example, it can be even used with explosives. This radiation is strongly absorbed by water, but I would say by all polar molecules in general, but it penetrates many dry materials like polymers, paper, textiles, and so on. And it is reflected by metals. So it has been proposed for many different applications. Probably the most famous one is for security reasons. You might have heard about the body scanner you can find in some airports. But it's also used for medical imaging, especially in dentistry, and also in pharmaceuticals, because it can predict the tendency of a drug to crystallize. It's used for the analysis of art objects. We all know that wireless communication is aiming to a higher and higher frequency, and it is approaching the terahertz range. It's also widely used in astronomy. But today I will tell you about food inspection, which is the topic of this meeting. So why terahertz radiation for plant imaging? As I said, it's a non-ionizing radiation. It's completely safe. It's non-invasive. So the measurements can be repeated in time and can give us a monitoring of the situation as time passes. It mainly carries information about the distribution of water because it's absorbed by water. But it also gives us information about what's inside dry objects like dry fruit, because dry biological matter is usually transparent with limited scattering effects, which is another important feature of this radiation. On the other hand, it's a relatively new technology. And this means that not many efficient sources and detectors are available, and those who are available are usually quite expensive. But this can improve in the future, of course, with technology development. What is more difficult to improve is the inherent low resolution, which depends on the long wavelengths of this radiation. This is our setup. It's a very simple and compact setup that can be portable even in the field. This, for example, is a picture of our setup. The black box is the source, which is simply a diode that works at very high frequencies, around, in this case, around 0.1 terahertz. And this metal square is the antenna detector array. That you can also see in the inset, maybe more clearly. with a leaf in place. So the setup is compact, portable, it's computer controlled, so everything is automated. So it has a lot of good advantages, but of course it has a low resolution, about 1,000 pixels. And this is 1,000 pixels in total, not 1,000 times 1,000. It's 32 times 32, okay? And the pixel size is in a millimeter range. So the resolution is really low, but that's inherent in this radiation, in the wavelength of this radiation. So with this setup, we could observe the leaf hydration status, which is representative of the plant health condition. We monitored in real time the water distribution, the hydration and dehydration dynamics, and also the spatiotemporal distribution of the water inside the leaf. But let's start with first, okay, I told you that this radiation is strongly absorbed by water, but is it true that we really measure what we want to measure, so the water distribution? So this was the first demonstration that the terahertz radiation, the terahertz absorption, which is reported in the y-axis in these graphs, linearly correlate with the water mass of the leaves measured with the gravimetric method. Of course, the gravimetric method is destructive. In order to make this comparison, we had to destroy the leaves. But after a calibration process like this one, then you can use this method to measure and monitor as time passes the water content of the same leaf, for example, at different times. And we demonstrated this linear correlation in 6 different species of different plants. And then we really tried to observe the water distribution in the different leaf compartments of a sage leaf. We focused on the midvein, and along the direction of the midvein, we extracted 7 different parallel directions from which we obtained this kind of 3D plots that shows the water content both as a function of time and of the distance from the PTO. And you can see, so we obtained these 3D surfaces that we were able to fit with a simple model that takes into account the diffusion process inside the leaf and the presence of chemical reactions that can alter the water distribution inside the leaf. Here in the bottom panel, you can see an example of the temporal behavior, which is an exponential growth in this case, and the spatial behavior, which has a sinusoidal behavior as predicted from the diffusion model. Okay, but this is kind of a basic science study. After these demonstrations, we moved to more practical applications, like the observation of the behavior of plants to ozone exposure. Ozone is anthropic pollutant, which is very, very important nowadays, unfortunately. So we exposed 2 different hornbeam species, Carpinus betulus and Ostrea carpinifolia, to 3 different levels of ozone levels, 2 different levels of ozone. Ambient level, 1.5 times this ambient level, and 2 times the ambient level. And in all these cases, we monitored the physiological parameters, like the stomatal conductance. And with the TeraHertz imaging setup, we observed the water distribution inside the leaf. So let's see the results. This is the result of the stomatal conductance for the 2 species. You can see in the top graph that Carpinus betulus at the highest ozone exposure stopped opening and closing the stomata during illumination and dark phases. while Austria carpinifolia continued opening and closing the stomata at all the three ozone level exposure. This correlates with the TeraHertz observations because this is an example of the images of the leaf taken in the TeraHertz region. From these images, we apply the threshold procedure to obtain the area of the dry spots inside the leaves that were not visible in the visible images. So we could identify some spots inside the leaves where the transmission of the terahertz radiation was higher. And this correlates with the water content of those areas. So we identified those areas as dry areas, and we measured the percentage of dry areas in both species. And here in the bottom panel, you can see the results. In Carpinus betulus, the percentage of dry leaf areas was not significantly different for plants exposed to ambient ozone levels and twice that level. But for Austria carpinifolia, we could observe a much higher percentage of dry leaf area in the plants exposed to the highest ozone level. And this is in agreement with the observation of the stomatal opening and closing, because we interpreted it in this way. Carpinus betulus, by closing the stomata at the highest ozone exposure level, protected the leaves from damaging, while in Austria carpinifolia, the continuing opening and closing and the photosynthesis activity led to damage, irreversible damage to the leaves. So we could observe the two species that are so similar from a genetic point of view, in practice react much differently to ozone exposure. Now let's go to dry fruit. As I said, we can also investigate the quality of dry fruit with the terahertz radiation. And let's start with chestnuts. One of the problems with chestnuts is that they can be infected by fungi, and so they can go rotten inside. And this is impossible to see from the outside because the skin is perfect. So you cannot tell if they are rotten inside or not until you cut and open them. And this is a big problem for industry, and this problem is increasing with time due to climate changes. So we used a sample of 50 chestnuts to see if terahertz imaging could give information about the health, the status of the chestnut inside. And so this is the distribution of the terahertz attenuation as a function of the weight of the chestnuts. We, from this data, we extracted the statistics of the attenuation over weight or over volume of the chestnuts. And we could observe a difference in the statistics between healthy chestnuts and rotten chestnuts. In particular, healthy chestnuts show a statistic that's more peaked around the central value, and it's symmetric, while rotten chestnuts has a much lower and wider distribution and asymmetric distribution. By choosing specific thresholds, we were able to select 30% of the unhealthy chestnuts that could be taken out of the sample while retaining almost all the healthy chestnuts. So although not perfect, this method could help in reducing the percentage of rotten chestnuts in a given sample. So we demonstrated this possibility, although the statistic was poor, because among the 50 chestnuts, which we supposed to be about 50-50 healthy and infected, in practice we only had 8 healthy chestnuts and 42 infected. This gives you an idea of how big the problem is for chestnuts producers. For hazelnuts, we investigated another problem. Hazelnuts can be bitten by the sting bug at an early development stage. And this means that the shell is formed afterwards. And when you collect them, when you harvest them, you don't see any hole. But if the hazelnut has been bitten, then it develops a very bitter taste that's not good for the human. Well, it's not harmful, but of course it cannot be used for industrial productions because the taste is really terrible. This is a big problem for industry because there are no automatic system to select good fruits from bad fruits, and the selection is still made manually by skilled workers who look at the hazelnuts one by one and select them. So it's very time-consuming and expensive process for the industry. So we propose to use TeraHertz imaging to select good and bad fruit. This time we work with 150 hazelnuts, out of which 110 were healthy and 40 were injured by the sting bug. From the TeraHertz images, like the ones you can see here on the left, we extracted a similar statistical analysis, similar to what you have seen with the chestnuts. And again, by comparing the two statistics, we were able to select about 30% of the healthy hazelnuts and cut all the unhealthy hazelnuts. So this is already a good result, but we thought we could do better. And in fact, this is a further analysis we performed. We built what are called the probability plots of the two distributions of the healthy and cimiciato hazelnuts, which are the blue and red dots. And then we took a sample of 50 unknown hazelnuts taken as validation sample. And we added each of these hazelnuts to the two distributions. And for example, for a certain hazelnuts, the result is the blue star and the red square when added to one or the other distribution. And we assigned the hazelnuts to the distribution with the shortest distance. So in this case, you can see the star overlaps perfectly with the healthy hazelnut distribution, while it's far apart from the cimiciato hazelnut. So with this procedure, we could analyze these 50 hazelnuts and build what is called the confusion matrix, which reports the true positive, false negative, false positive, and true negative rates. And we could detect, we had a true negative rate, a perfect true negative rate of 100%, and 0% of false positive rate, which means 100% specificity. And this is a very interesting result. Okay, and this was done with, you can see these very low resolution images. So let's imagine what we could do if we could improve the resolution of our TeraHertz images. So we are moving towards high resolution, which means moving to higher frequency. Still in the terahertz range, 2.55 terahertz, we have a quantum cascade laser as a source, which is a highly engineered laser source, but it has some problems with interference effects, but we were able to solve these problems, and this is an unpublished result. This is the transmission of a leaf with this high-resolution imaging system. You can see the improvement in the spatial resolution of the image. And this, remember, it's not like a visible picture, the radiation reflected by the leaf, but it's the radiation that is transmitted by the leaf. So it brings information about the water content inside the leaf. Okay, so let's go to the conclusion. We demonstrated that the terahertz radiation can be used as an effective technique for food and plant imaging. It permits to monitor the spatiotemporal behavior of water inside the leaf and can observe the response of the plants to environmental stresses. And it's also good for dry fruit quality assessment. In the future, we're working on developing this high-resolution TeraHertz imaging system and use it to monitor the response of plants to pollution, to heavy metal exposure, and these kind of things. And also use artificial intelligence combined to TeraHertz imaging for dry fruit assessment. Let me finish with by thanking all my collaborators of the University of Pisa, of the Italian National Institute for Nuclear Physics, and also CNR, the Italian National Research Council in Florence and in Portici near Naples. And thank you all for your attention.